Thank you so much, all of you, for coming to the Center for Eudaimonia and Human Flourishing. I can't tell you how much of a pleasure it is to be able to welcome Jean-Pierre Changeux, who is without a doubt probably the most eminent neuroscientist in the world. His work on neurotransmitters, and specifically the acetylcholine receptor and the nicotinic receptor, is legendary. This happened back in 1970. And I think was really the first characterization. And throughout his career, which has been basically spent at the Pasteur Institute, Jean-Pierre has done a lot of work, both at the molecular level, but also famously, as many of you will know, on consciousness. He's, together with Stan Dehen, he's come up with one of the most influential theories of consciousness called the global workspace, or the neuronal global workspace, full of incredible ideas. Now, one of the things I truly admire about Jean-Pierre is the fact that he has always just been about the science. He don't care for all of these, all the extraneous things that take up so much of our times, running things and so on. He just cares deeply about the science. I had a good demonstration of this. I met one of his collaborators, a young man called Guillaume Dumas, who was just with some of the people in from the center, was in Corsica at something called MESET, which is about consciousness, really. And he most unexpectedly came back with you to Oxford, and I sat there and we started talking about how he met Jean-Pierre, with whom he will present some of the work you did together. And he said, yeah, we met at the coffee machine. And then I realized who he was, and then we went into his office, and then we just started doing the science. and I think that's joie de vivre, this idea of really what matters is not who you are, how old you are, but it's the quality of your ideas. And I think for me, that's how I would like to continue my scientific career. So in many ways, I model myself on Jean-Pierre, but to actually be like Jean-Pierre is almost impossible because, of course, on top of doing all of this work, he has also held many other positions, including being one of the people that decide with artwork which one should be, as part of the taxation that happens when you die in France, which one should be given to the French state. So he's also an expert on art. But that's not what he's going to talk to us today about. He's going to talk to us about what makes us human. What is it about the humanization of the brain, all the way from genes to cognition? And I don't want to spoil his fire, but I think there will be fire. So please, Jean-Pierre.
Thank you very much. Thank you very much, Martin. I must say I am extremely pleased to be here for several reasons. The first is Martin. We know each other for decades, and has always been very constant friendship and admiration, I may say, for what he has done. And the other reason is I am here with students and collaborators of Morten who are on the way to become professor or are not yet professor. And this is, for me, a very important public. Why? Simply because what I am going to talk about is mainly a work for the future. I'm sorry to say that, and I'm going to disappoint most of you because I am not going to solve the problem of the hominization of the brain. But what I would like to do is to suggest a number of avenues where you can walk and make discovery and contribute to the understanding of the origin of man, but I would say humanity in general, and in particular the brain. So the title is Hominization of the Brain. How did our brain become Homo sapiens, that of Homo sapiens? From genes to cognition, which is very far from each other. And that's precisely perhaps the point I want to emphasize from the start, that many of us are either molecular biologists or cognitive scientists or computational scientists and so on. And we have to realize that to understand the brain, you have to understand the genes, you have to understand the behavior, consciousness, and everything which is in between. And what is in between is enormous, gigantic. And that's precisely the difficulty. And if you want to progress in difficult system and complex situation, you have, of course, to simplify and to follow a certain number of ideas. And that's what I am going to illustrate today. I don't want that you believe that I am going to solve the problem. Again, I repeat myself, but this is important to know. So what is the human brain? You know about it. These are pictures from the 19th century of the brain, and 1.4 kilogram complex circumvolutions, and made up of neurons, and about 100 billion of neurons, and about 1 million billion synapses, which makes the human brain one of the most complex organs of the body. Now, of course, these neurons are not distributed randomly. You already see in these 19th century pictures that there is a high organization of the brain, and you may specify it more closely. This is a macroscopic level, and this could be a mesoscopic level. That, for instance, if you look at the cortical specialized areas, then there are about 180 per hemisphere from the work of Bonnes and others, and about 1/3 of them are new compared to what is present in the monkey. Now, you can also see from this drawing from the wife of Stanislas Dehaene, Ghislaine Dehaene-Lambert, that if you compare the long-range connectivity of the newborn with that of the adult, you find that they are almost present already there. So it means that a lot of this brain organization is innate. It is species specific, I would say. And therefore, we have to really understand how this species specificity develop. And also, you all know that we are highly diverse, variable, with different cultural background. There is a diversity that we have to account altogether. So this is a very interesting issue. Where do we come from? Well, of course, some of the older ancestors of Homo sapiens are Australopithecus here, and you have in about 2 million years a dramatic increase of the size of the brain, which is here 500 cubic centimeter to 1/4, 1/5 cubic centimeter. And so in about 2 million years, which is a very short time in the evolutionary scale, the human brain originates from that of Australopithecus and Homo habilis by more than the doubling of the size. So we have to account for that. And the other thing is that not only you have an increase of brain size, but of complexity of organization. And if you compare here the brain of the Paniscus to that of Australopithecus, to that of Homo habilis, and then Neanderthal and Homo sapiens, even though the colors are somewhat different from in their meaning from these different pictures, but you can already see that there is a complexity which is developing, that some territories are becoming more important. For instance, from Australopithecus to Homo habilis, with the prefrontal cortex in particular, which developed, and the Broca area. So there is an expansion and a bulging of the brain, which is becoming evident now with Homo sapiens. And you can see here from Neanderthal to Homo sapiens that there is a very significant change of shape. There is a global compaction and expansion from Neanderthal to Homo sapiens, which is taking place, and the development of the prefrontal, temporo-occipital cortex, and the cerebellum. These images are mostly from Jean-Jacques Hublin, with whom I work for several years now. The anatomy of the brain is changing, evolving, and the behavior is dramatically changing. The human brain develops cognitive abilities, as you all know, which are quite distinct from those of non-human ancestors, in particular the chimps, which we can follow the behavior, but not from Homo habilis or erectus, except from the cultural object I will show in a few minutes. So the main differences include language, self-awareness, theory of mind, which is a social relationship, symbols, creativity, and also cultural evolution. And this is not an exhaustive list. It's simply a few of the features which make the distinction between human and non-human primates. So we have to understand that there is different levels of behavioral abilities and levels of organization of brain connectivity, which are already present and that we have to relate one with the other. Now, one of the most interesting aspect, which is often forgotten when one speaks about human evolution and the selection which led to Homo sapiens, is that very early on there is a culture which began even before Homo sapiens and perhaps even Homo habilis. So there is already a cultural development, apparently in the case of Australopithecus, surely with Homo habilis, with Homo erectus, with Homo heidelbergensis, and Homo sapiens. And you can see that the cultural evolution is also changing. If you look at the tools, for instance, in stone, then the first tool found around 3 million years in Ethiopia are these very awkward kind of stone fragments. Then there is the apparition of the biface with Homo erectus, and this is an intentional symmetry. So it means that there is already an aesthetical aspect of cultural evolution, which is beginning at that time. And then with Homo heidelbergensis, the first funerary situations are being discovered, not only sepulture, but also offerings, which are associated with the sepulture. And finally, you have this elaboration of these beautiful cave paintings. And what is fascinating is that you have already all the characteristics of the contemporary art already 30,000 years ago. I would not say that we have made much progress in that time. Sorry to say. Of course, this is associated with the development of language, of course, but I would put art and language in parallel for the very origins of the human brain. As I said, with Homo erectus, I don't think he was speaking, but maybe he was gluttering or whatever you say. But there were already a sense of aesthetics very early on. So there is a clear relationship between the biological evolution and the evolution of culture. This relationship has to be further investigated. I just show it very briefly, but of course you have to document that much more than it's being done. And I would like to raise at this point the main paradox of my presentation. And the paradox is that if you wish to compare genome with brain evolution, you have to realize that when you go from yeast, of course, they don't have much brain, but from worms, 18,000 genes, flies, 13,000 genes. And when you go from fly to mouse, the number of neuron is increasing by more than 2 orders of magnitude. And when you go from mouse to human, you go for 40 to the 6 neuron against, as I said, 100 billion of neurons. So it means that at this level, there is a non-linearity between the evolution of the number of structural genes and the number of neurons. So many of the neuroscientists don't care. I'm sorry to say, they work on the human brain, they work on the monkey brain and say, well, it's okay. Well, it's not okay for me. I am dramatically puzzled by this paradox, and I think this is, if I may say, the most interesting question for the new generation to solve in the future. And this is further documented by the work of Guillaume Dumas, he did in Paris, was to look at the closer evolution of the coding protein sequences, compare between chimps and humans, for instance. And the most conserved sequences are in the testes. So the women who are present here may be afraid by that. It's a joke. And then among the other proteins, the liver, the kidney, and the lung are also very well conserved. And when you look at the brain protein, they are less conserved and like the ovary. I'm sorry for this kind of comparison. But the interesting situation is that the sequence of the brain protein is not so well conserved, and this is really a paradox. And again, the less, I'm sorry, I may have said the opposite, the less conserved are the testes, and the more conserved are the brain proteins. That was the lesson of this investigation, that the conservation of brain coding protein sequences is dramatic. So this increased the difficulty for us to try to understand how the evolution took place at genome level. So again, in summary, there is a similar small number of structural genes in mice, chimps, and whole human genome. the brain protein sequences are highly conserved. So how these things evolved? Yes, through genetic regulatory events. So that's the conclusion we have to say. There might be some local evolution at the level of the structural gene. I will mention most of the data at this level, unfortunately. But in fact, what's happening is essentially at the level of the regulation of gene expression. Now, to try to solve this issue, we met together with Klaus Ingetag from Hamburg and Gulas. And the question which we raised was trying to propose an intermediate hypothesis, which we call a connectomic hypothesis. It's not really from cognition to gene, it's in between, precisely at the level of the connectome to genome versus connectome, and said that the central dogma of genetics that one gene encodes one protein, which itself encodes one phenotype, is wrong. It does not hold for complex function, particularly the cognitive ability of the brain. You cannot say there is one gene changing, there is strictly one function which is changed. And this is, of course, dramatic in the case of psychiatry and neurology, where you have most of the time single genetic event and a change in the behavior associated with some kind of disorganization or reorganization of the brain. So this clearly shows that there is an issue of trying to relate the action of genes to how the connectome develop in the brain. So the proposal we make is that, accounting for the conservation of the sequence, there are some genetic regulatory events which may actually take place in non-coding sequences, and that in fact these events are having as a consequence a control of what we call connectomic fundamentals, which are some kind of units within the connectivity of the brain, which could be under the control of the genetic regulatory events, but which are at a lower level of complexity than the whole brain itself. So this is some kind of view which is, of course, a problem to try to solve. Is how can we relate some regulatory events in the genome to some modification or control of connectomic fundamentals. I think this is the main issue. I am not going to solve it. I am going to, again, show you some trends of how to approach this issue. The other difficulty and final difficulty is that if the central dogma of genetics which account for protein, of course, but does not account for the brain functions, then what's going to happen? In fact, there is, as I say, a very small number of structural genes, 20,000, and nevertheless, the proteins are coded, even the brain protein, by this sequence. So we have to be in a situation where there are some kind of relationship between the gene in the genome which control the expression of gene. This kind of interaction is completely not understood at this stage. So we call it an omnigenic concept in the sense that to code for the brain, many genes of the genome have to be involved, and some of them within this omnigenic background are going to control some connectomic fundamentals, and perhaps we can try to sort out from the genes which control the connectomic fundamental what could happen at the level of the omnigenic kind of environment. And we should say that this an issue of omnigenic complex is present in immunology as well. It's not exclusively in the case of brain science, and up to now it's not solved. So I am not going to solve it today with you, but I just want to say that there is here an issue which, as I said at the beginning, is fundamental for the molecular biology of the brain. Now, let's look at some connectomic fundamentals. I think several of you are going to find them trivial. They are, but if they are under the control of some particular gene, then it would be helpful. One is the increase of global brain size, the total number of neurons I mentioned already. Second is scaling up of the cortex and multi-level organization up to conscious processing. The third one is something that you may not realize, is a shift of cortical reafference from lower to upper layer in the human cerebral cortex. I will explain that in detail when I am going to talk about that. Now, there is another one which is extremely important, is an extended period of postnatal development where epigenetic processes of synapse selection are taking place, and which level culture may emerge even if ancestors of Homo sapiens. And last but not least, in this list of 5, of course there are many more, but I selected 5 important of them. There is what I may call the late postnatal dendritic expansion beyond age 2, where the oral language develop and the written language, and after all, science and ethics and arts, whatever you wish, within the cultural background development. So again, what are the genetic elements for these 5 connectomic fundamentals? So first is the increase of global brain size, with, in fact, correspond to an extended prenatal, not postnatal, but prenatal period of embryonic development. And this is indeed something which is happening here. You have an increase of the number of brain neurons from the macaque to human sapiens, 87 billion neurons compared to 6.4. And during prenatal development, you have also a change which is not very big, but quite significant. 35 weeks in chimpanzee, 37 in gorilla, and 38 weeks in humans. So you have to realize that a small number of weeks of embryonic development before birth is enough to give such a kind of change in the global brain size. And most of all, it's not the brain size, but the cortex, the cerebral cortex, is developing in a spectacular way, and therefore cognition, as all of us know. And I will come back in my last slide to this particular, remember this particular slide. Okay, so here we have a nice connectomic fundamental, which is the increase of the total number of neurons. And this has computational consequences, and this has been done mainly by close-signal work. And as you know, if you have neurons connecting with each other, you can have a full connectivity, which is taking place here, and only a limited one, which is near constant node connectivity, you know, and this is maintaining the proportional connectivity between neurons. The more connection they are made, the more connection, and it's not, as you shall see, not the case. The more neuron you have, to some extent, the connectivity does not follow. And this can happen because they are with this kind of near constant node connectivity, possibility to create some kind of sparse connection between groups of neurons, which is not taking place in smaller brains of macaque, for instance. So there is a more local connectivity, which lead to that we call network sparsity, which is, of course, interesting because you have here a way to diversify the neuronal networks that, of course, you don't have in computers, but all the network is the same. So now this is a interesting aspect for diversification of function, and in particular, the acquisition of the lexicon. Now, there is also another computational consequence of the increased number of neurons with this near constant node connectivity, which is the form a larger association than this one, which are called module. And the computational analysis of neural networks with modularity shows that you may have an interesting properties which appear, which are not present in networks without this modularity, which sustained activity, enhance working memory. And this is just a computer experiment to show that there is enhanced working memory with modularity, which is taking place. So one should realize that even in computer models of the brain, at the beginning, one should not deal with some kind of homogeneous hyper-connected network, but with a network where there is, again, constant node connectivity and sparsity and modularity. And you can have, in fact, associated with a simple phenomenon, which is increase of the number of neurons, some cognitive phenomena which are appearing by themselves without the need of any additional gene expression. So this lead to an interesting conclusion concerning the genes involved. Of course, you are starting with human with a brain which develops through steps which are very similar with the other primates, from the early embryo up to the, let's say here, 5 months fetus. And clearly, you have already in the human brain formation a background of all the genes which are involved in the mammalian brain. So for hominization, we are not going to count in genes. So these mechanisms are common, and for hominization, we need more. So there are some human brain-specific events which are responsible for this extension of the prenatal embryonic development, the ZAC gene, which regulates intrauterine growth, and especially those which are involved in humans, and the most of all, interesting one, which is the microcephaly gene, which has been studied by the group of Mu Mingpu in China. And he can have overexpression of the human gene in macaque. Well, I think these experiments may be debated from an ethical point of view, of course. But the fact is that there is an increase of brain size and gray matter and working. There is a myelinization taking place, which is quite characteristic of the human brain. So you can confer through the transgenic insertion of this and increase the number of copies of this microcephalic gene, some character of human maturation. Now, I don't see the paper. This was done after a few months. What happened after? Nobody knows. So I am not sure that this is going to be the complete model, but at least you can see that you have example where, again, from structural genes, which are acting as regulatory genes, all these are transcription factor genes, for many of them, you can have this kind of effect. Now, the second connectomic fundamental, in addition to the increase of the number of neurons, as I said, is the scale-up of the cerebral cortex. And it's a multi-level organization. This has been discussed in this laboratory for years, and it is an issue which is, of course, essential for the understanding of the evolution of the human brain. So the computational examination of the development of the cerebral cortex from mouse to macaque to human has been interpreted by several groups, including ourselves, as being due to the increase of the number of layers of organization. This is something obvious to realize. Of course, in the mouse, there are only a few or one level of organization. And if you go to the monkey and up to the brain, you increase the number of level of organization which are nested together into hierarchical organization. And the interesting thing is that in this low level kind of organism, you have mostly short-term connection, like in the mouse and in the macaque, but long-range connection are already appearing in both of them. But the thing is that there is an increased contribution of the long-range connectivity, or which is called periphery, and with the highest level, we can see the development of long-range connection. And this has also been, as I said from the beginning, studied by computational investigation of, in particular, Krubitzer and Selke, where you can see that increasing the number of level of organization, of layers or levels of organization, then you have an enhanced computational ability and a decrease of the number of error in a particular task. So it means that from this simple-minded hypothesis to have increased level of organization compared to simply the whole size of the population of neuron present in the brain, you may have an issue where the relationship between the gene and the cerebral organization is taking place without a cost of gene, which is very high. And of course, the idea is that with this increasing level of organization, the number of long-range connections develop and contribute to the white matter, for instance. And that's exactly the observation which actually I took from my course at the Collège de France, and in the discussion we had together with Stanislas, the idea that from Barr's early model of a global workspace, without any kind of plausible neural organization underlying, we could propose that this long-range connectivity, which is identified in the cerebral cortex intramonicaural and Desjardins in the end of the 19th century. So this long-range connectivity, which is also found by Patricia Goldman-Rakic in the monkey between the prefrontal cortex, the parieto-temporal cingulate areas, and which has been confirmed by the group of Kennedy in their study of the connectivity of the cortex, where the strongest links, which compose 24% of the cortex, are those which we have postulated in the global neuronal workspace. So the idea being that when you don't have access to consciousness, which is a subliminal kind of stimulation, I hear for visual stimulation, you have a feed-forward propagation until it reaches the global neuronal workspace, where then you have a sudden invasion of the whole cortex through a feedback connection and a reverberation between the different territories of the global workspace. This is illustrated by the model we propose with essentially Stanislas, that you have the feed forward, the feedback, and when the access to consciousness takes place, there is a phenomenon that we call ignition, which is taking place. Ignition meaning that there is some kind of burning up, which is a bad metaphor, but it's of the cerebral cortex taking place during conscious access. So again, this is some kind of experiment that are very classical from the work of Stanislas, where you can develop what is called a masking task. Here you have the word lion in between two empty screens, and you are here the word note between two heavily loaded screens. You see the difference between the two? So this one can be read, this is not read in this task. So when there is visibility, global workspace, some elements of the global workspace become activated, and otherwise in subliminal vision, then there is, of course, the visual areas which are going, and the same thing for detecting a sound, and of course a musical sound included, which again follow the same kind of rule. So not only with this simple-minded kind of hypothesis that long-range connection through reciprocal connection through different territories of the brain interact with each other, lead to a quite interesting kind of prediction in the performance of cognitive function. This just to illustrate that brain imaging, which I am always quite doubtful about the data, because they are interesting, but they are, of course, playing above a spontaneous activity which is very high, and that is most often ignored by the quality is ignored, the quantity. So I think it's good to have some electrophysiological data at the cell level, which were obtained by Rolf Sema, and you see here subliminal stimulation of neuron from the visual V1, V4, and then when access to consciousness takes place, there is this ignition that you can see, which corresponds to a broader, long-lasting kind of activity. Now I'm pleased to say that reading the work of Morten and colleagues, I was very happy to see that he was going a step further, which is not only to have evidence for ignition, but and several territories being activated, but that the newborn make a difference between cute and uncute, unfortunately, faces of children. And this is interesting because it means that you may have some features which have to be understood, which are characteristic of the aesthetic ignition compared to a regular kind of ignition. And that's where the rules of art, which I discussed extensively in the past, may play a role. So we may say that ignition can be a signature of conscious perception. And the question is, how do you get there? Simply, again, for this second hypothesis of the multi-level development of the multi-level organization, which is scaling up to the global neuronal workspace. So there are some candidate genes, which has been quite well studied by the group of Van der Hagen in Brussels, in Louvain, and they have identified some regulatory genes here, which are selectively involved in the cerebral cortex expansion, in addition to what I said before, which is Notch2, and these other SRGAP2 or RHGAP2. What is interesting is that it is not, in the case, a necessary change in the gene of structure itself, but that could be a duplication of the genes. And that's, again, you explain the parsimony, which is part of the paradox. And he has noticed that the copy number of Notch is 4 in human and only 1 in chimps. So this is a strong argument for having this kind of genetic control taking place in the course of human evolution. I don't want to say that we have explained everything, but at least it's one argument from the genetic point of view and one possible mechanism among many other ones which may take place. Now, I mentioned the shift of cortical layer reafference, and you may ask, what does it mean? But it means if you look at the cortical layers in rats and human in detail, which you don't see on these slides, but if you carefully look at the soma size of the neuron of the cerebral cortex, you know that there is below layer 4 called infragranular, and above you heard the supragranular layer. And you can see that the size of the cell body is in fact decreasing from infragranular to supragranular in the monkey, but you find a shift, which for us is dramatic, from supragranular to infragranular in humans. So there is a real balance. And of course, you know that the supragranular are neurons with long-range connection. So you can make the relationship with the global neuronal workspace. So this is a very interesting phenomenon. Unfortunately, we don't know the genetic of it. Yes, I hope that the new generation of scientists will actually give an answer to that. Now, the fourth connectomic hypothesis I want to emphasize, I may stay a little bit of time on that, is the extension of postnatal development. So when people compare the human brain with the monkey or the cat or the rat or whatever, they don't realize that the adult human brain has developed for about 17 to 18 years, if you consider that an 18-year person is an adult, whatever, for psych. This is to be discussed now. But clearly the 14 and 15-year-old are not adults. But so it means that there are a lot of processes which are taking place during this long period of time, and the human beings are the only one when they appear on Earth to spend perhaps 1/3 of their life, or maybe half of their life, because they were the Homo sapiens at the origin were living for 30 to 40 years. So nearly half of the life of the adult human is making the brain. So you can realize that there is a development of brain complexity which is taking place after birth, which have contributed to the selection of the human brain to be used only a small number of years. It's amazing how you have that. So what about this postnatal development? But of course, if you look at the newborn brain up to the adult, you have an increase of 5 times for the weight or the volume, which is a big factor. Even if the total number of neurons is not changing too much, but there are some increase, some decrease. But the number of neurons, the glial cells, of course, are proliferating a lot. But you have a dramatic increase of size, which is due to the connectivity, of course, to the development of the connectome. So it means that about 50% of the synapses of the adult brain are developing during this postnatal period of 15 to 18 years. And it's clear that this is really characteristic of human. In chimps, it's 10 to 15, 7 to 15 in orangutan, and in rhesus monkey, much smaller, 3 to 5. So it means that there is a dramatic increase of synapses taking place in the brain after birth. And if you count the total number of the 10 to the 15 synapses or half, and the number of seconds to reach 18 years, you can compute that there are about, this is a very rough estimate, about 1 million synapses per second being made in the brain. So it means that if you are doing experiments with newborn, be careful, because in a few hours, the number of synapses have changed quite significantly. So this is just an image to illustrate the arborization of cortical neuron pyramidal cell from 3 to 15 to 24 months. So there is a dramatic development, which is of the brain, of the connectome, which is taking place after. So it's really a connectomic fundamental. Now, what's happening during this period? Of course, there is an increase of synapse, the layers over layers, the levels over levels are developing postnatally. And the idea we propose with Antoine Danchin and Philippe Courage in 73 is that you may have some events taking place successively all over the development and sometimes mixed with each other, where you have overgrowth of nerve connection to the target, and then a selection of synapse taking place with the elimination of some connection, as you can see, which has been called pruning by the American language. So this is a mechanism which is some kind of selection, which can be compared by Edelman with Darwinian mechanism, whatever, but except that it is epigenetic. You don't need change. And also you may have for each of these steps the same kind of events taking place, and therefore genetic control taking place, which is first of all the proliferation of synapses. I already mentioned that before. Then the selection, elimination of synapses, which is taking place, and resulting an important phenomenon taking place is if you have neural activity taking place at this critical stage, then there is a control of the selection which is taking place. So here you have a proof of that, in the sense that if you block by tetrodotoxin the connection between the optic nerve and the thalamus, then you can see that if you introduce tetrodotoxin, then you have more connection than if you let the system evolve spontaneously. And this is very significant. Tetrodotoxin blocks the function, increase the number of synapses, because it blocks the epigenetic selection, which is taking place at this stage. So this is, of course, an interesting feature as far as a connectomic fundamental, that first of all, if you have prolongation of the postnatal development, then you have more and more waves of synapse growth and selection, using each time nearly the same kind of genetic equipment to have this selection process to take place. So we can, of course, simulate this kind of situation here in the acquisition of rules. I call epigenetic rules for art, but also for task, which is a Wisconsin card sorting task. Of course, a naive brain have not learned the rules. So you have to, first of all, to ask the subject to learn the rule, and then ask him or her to apply it. And here is just to illustrate that the reward in this model has been postulated to contribute to the selection. And you can see the importance of reward in synapse selection, which again is manifested by a decrease of the total number of synapses. Okay, so this, of course, has very important consequences for us. First of all, Desjardins in 1901 showed that a posterior lesion of the cerebral cortex here cause alexia without agraphia. It means that reading is mediated by this set of connection and not writing. Just to illustrate that reading and writing have connectomic substrate present in the brain, which develop epigenetically after birth. And here, early slide of Martin Ingvar, which was really pioneering the work with Castro Caldas from Portugal, where they compare illiterate to literate subjects. And you can see that in the literate subjects, you have a different connectivity in the prefrontal cortex and other places. This has been confirmed by the group of Stanislas and further documented. And our view is that there have been some epigenesis of the developing connection postnatally, and there is some critical stage of activity for the acquisition of written language. Now the issue of synapse elimination here is of course very difficult to get. My hypothesis is that there is a synapse selection which is taking place there. And of course, you have the development of culture and the diversification of culture, which is, of course, epigenetic, and many other aspects of language I am not going to talk about. It could be a full seminar just to look at the epigenetic aspects of language acquisition. Now, I am going to show briefly candidate genes, and then there are plenty of genes which are involved in the extension of post-synaptic development, which are involved in the growth of the human body in general, not only the brain, but the rest of the body. And there are some genes which have been identified for, again, by Van der Hagen, for synaptic plasticity, which is the selection system which I just discussed. One gene here, it's again. And the most interesting situation is, of course, autism, schizophrenia, and depression, I think, where, again, there is clearly an altered maturation of the brain, which is taking place at early stage in the case of autism, just at the time of acquisition of reading and writing before, and in the case of schizophrenia, of course, much later. And now when you look at the predisposition to autism and schizophrenia, you find there are at least 200 predisposition genes. Yet, an interesting aspect is that 5,200 genes of these 200 are for synaptic components. So it means here again that we are in a paradox in the sense that we want to save genes, and indeed we can save them through the extension of the postnatal development, or through the synapse selection rules, which are common. But now we are in a paradox that yet is not solved. And the idea, the general idea, is that I always like to say that gene network code for neuronal networks, which is a little bit caricatural, but means that we are facing this omnigenic context I just mentioned. I will end now with the fifth hypothesis, which is the expansion of dendrites beyond age 2. So this is again something which is clearly highly developed in humans, and here is the extension. This has been developed by a group from Slovenia or from Bulgaria, from Yugoslavia, from Petonček, from Zagreb, which is where is Zagreb? It's in Croatia. Thank you. So from Croatia, and they have made very interesting observations. They have been working with Paskourakis before, and looking at the expansion of dendrites, basal dendrites, in the pyramidal cell of the cortex from layer 2. And you can see that after layer 2, there is a dramatic expansion. Yeah. And the idea, of course, is that this corresponds to the time of acquisition of language. And the hypothesis would be that there is this additional step among the kind of genetic events taking place during hominization, which is this increase, late postnatal increase of dendritic carbonization, which is taking place, and this is my last data series, which has published in the book of Federici, and where you see that she claims that when you look at brain imaging of Chinese, English, and German-speaking people, you find some differences. Now, this has to be confirmed and further documented, but at least there are some differences, in particular between Oriental and Occidental people as far as oral language and written language as well. So, what are the regulatory events involved? Unfortunately to say. So this is a summary of what I wanted to say, that, we have increased brain size and number of neurons and some genes involved, scaling up of the multi-level organization, some genes involved in addition to the CF1 involved, an extended period of postnatal development just discussed, a shift of cortical layers, which contribute to the development of long-range connectivity, and then this postnatal dendritic expansion. So where language acquisition took place, well, it's difficult to say. It's divided between Erectus and Sapiens. So I will close with a part which may not please Morten and be somewhat in contradiction with the title, the name of this building, to some extent, is that if you look at just what I said, of course, I've been thinking mostly about the cerebral cortex, not so much about the subcortical areas. And if you look at the, at least from these data, anatomical data for the expansion of the cerebral cortex compared to the non-cortical area, you find that there is a non-linearity between the development of the cerebral cortex and the subcortical areas, which are involved in social relationship, the theory of mind, and so on and so forth. So it would mean that there is some kind of disharmony in the evolution of brain, and this is from the work of Holmes. And as you clearly see, there is a divergence of neocortex compared to striatum, diencephalon, and so on and so forth. So the question is whether there is a disharmony between cognition and emotion and social life. And I may say that when you see what's happening today in the world, you have a situation where in Gaza, in Ukraine, and in many other parts of the world, globe, there is something happening that we don't anticipate from a harmonious development of the brain. So this was mentioned already by Konrad Lorenz. And I will close with one quotation from the philosopher Günther Anders, which in 1956 wrote a very interesting book, The Obsolescence of Man, the Promethean gap between our faculties of fabrication and imagination and the enormity of what we can produce and set in motion with our brain. Meaning that we are not thinking enough, in particular in the political circles, between the ability to have a harmonious life and the knowledge we have about the world through our cognitive ability, science, and technology. So that's where I think we need some universal ethics and aesthetics. Thank you very much.
How wonderful. Thank you, Jean-Pierre.
Sorry for being a little bit critical at the end.
I think you must be. This is why we're here, yes. So I'm sure there are many questions.
Yes.
I think we'll start here, and if you could monitor what we... No, I can monitor this online. But Jamil, please start.
Yeah, absolutely amazing tour de force of what it is to be human. I'm particularly interested in this idea that long-range connectivity, increasing long-range connectivity, seems to have been in the second and in two of your 5 criteria. Does that mean that the quality of consciousness, there's a kind of almost a discontinuity in the phylogenetic chain in quality of consciousness? So for the global workspace, in a sense, is qualitatively different in humans as in chimps or something like that, because it seems like there isn't that modularity and long-range connectivity between those modules from what you're describing. So the hypothesis is that there's a...
Yeah, I think there is, you are pointing to a very interesting point, which is the contribution of long-range connectivity to the shaping of the brain. And curiously, when you look at Neanderthal brain, remember the slide? And Sapiens brain is more globular. And the idea we put forward is a very, very, very speculative idea, is that the long-range connectivity would contribute to the making the brain more globular. This hypothesis had been proposed quite a while ago by other people. I did ignore because we thought we were original, but we are not. And but the idea is interesting, but it's still very hypothetical. And that the long-range connectivity not only would have a functional role, but also a structural role in the development of the connectome.
But would there be a role in the shift in the phenomenological quality of consciousness?
The idea we have written with Michele Farisco a paper about is artificial consciousness plausible? Contribution of brain knowledge. And the idea is that you cannot use the same word when you are looking at human consciousness and monkey consciousness. You cannot, because there are a lot of qualitative differences between what we can do. So when you talk about use the word consciousness, you should say consciousness in the monkey, consciousness in Drosophila, and consciousness. If you want to use consciousness or don't use consciousness, use some specific conscious processing phenomena. So we say that explicitly, that there is a misuse of the word consciousness, going from God to nicotinic receptor, which create a lot of confusion, while the situation is not confused. And it's clearly there are differences between the monkey ability to process conscious representation to that of human. The human consciousness is much richer than that of the monkey, of course. But it is, I think, an epistemological mistake to use the same word to describe what's happening in Drosophila and what's happening in. Yet, you may have some simple-minded features which characterize conscious processing that you have to get. And for instance, you may say the masking task is a criterion. Now, do you have a masking task, or do you have the trace versus delay conditioning task being performed in Drosophila, monkey, and human? Yes, you do. So this is some kind of universal criterion, which is part of a much more complex system I would use. Is a system view of conscious processing that you can find in human. So never use the word consciousness between mice and humans, except if you mention this is mouse conscious processing or human conscious processing.
Can I maybe just use my provocative chair?
Please go ahead.
So we discussed this earlier, there's something very interesting about time in the brain and time critical processes, because of course, the main aim for all brains is to survive. And there are quick decisions to be taken, there are things to be approached, things to be avoided. And of course, one way of thinking about long-range connections is that they confer the ability to broadcast signals, to make something, I'm not going to say conscious, but at least available to a given system, yes? And so there's something about the criticality, something about the avalanches that can happen in time for you to make the critical decisions. And it is curious, I think, and you mentioned it in your talk, that the number of, because the general principle of brain wiring is the EDR rule, the exponential distance rule. You connect with those close to you, and then there's an exponential drop-off. And yet, those exceptions to that, as shown by Henry Kennedy, the long-range connections, the proportion of those increase in Homo sapiens compared to chimps, compared to macaques, compared to mice. So it's almost as if there are fewer of them, and therefore, this is what confers us with these abilities to do this selective
I
process you
think
talk about.
I agree fully with you. Now there is, for the long-range connection, a debate. I want to say,
Yes.
I don't want to hide things. And the way you define them, and also the contribution of nearest neighbor kind of interaction between which could, at the end, give a long-range interaction, even through nearest neighbor interactions.
Yeah.
So that's you have to realize that what we wanted to say, of course, is based on anatomy, and but there might be additional ways of creating long-range connectivity functionally. That's one thing. The second thing is that, of course, the processing of conscious information depends on the speed of propagation. And there is, of course, a myelinization, which is taking place.
In the long range, which is not present if you go from...
Well, which is not present at the beginning, but at the few days after birth, and which is increasing progressively with time. So, Ghislaine de Van Lambert have looked at newborns, but when she said newborn is either a few days or 3 months, so it's in fact quite different, but the story is true nevertheless. It is that they showed that they can find a masking task, the newborn, but with a time scale quite different from the time scale of adults.
Absolutely, because they have to have that longer... Yes.
So it's just a fact which goes along with what you
And
said.
of course, the prefrontal plate is not quite developed and so on. Anyway, I'm sure there are other questions. Ramon? I think.
So when you compare the complexity of the genome with the connectome, you could look at the number of genes versus the number of neurons. But if all the genes are like very heterogeneous, and many of the neurons might be very similar, and there's a much smaller number of types of neurons, is there like a better way to compare the complexity of the two systems so you don't get this inconsistency where the number of genes remains the same, the number of neurons goes up, if the actual complexity of the types of neurons you're creating with these genes remains the same as well, and it's just the number of them that's increased? Is there any other way to compare?
So what is the question?
Is that actually a fair way to compare the complexity of the two systems?
No, I'm caring with that. Of course it is, but it's no. I did not want to exhaust all the difficulties, which are. I pointed to a number of big difficulties, but there are many more, as you just say, and let's say the biochemical diversification of neurons, which maybe is close to what you mentioned, is something which we don't know. We completely ignore. And it can be mapped now with messenger RNAs, as you know. But in addition to this complexity along your line, the synaptic complexity above it is even larger. And nevertheless, you see brain imaging for reading and writing. So what does it mean? It means that in this brain image, the epigenetic kind of selection or buildup of the brain connectivity is dominant. Because why would you see some brain imaging using traditional fMRI technique? And I remember I showed to Stanislas this work of the Swedish group. You say, oh, he was not so happy. I said, well, why don't you do it? And he actually did it with the same subject from Portugal, and the Swedish group, and he confirmed that is true. So it means that when you are looking at a brain slice it is the brain slice of an individual, and you don't expect to have the connectivity fully reproducible from one individual to another. I may say that I should have said that, but I usually talk about that in ethical issues, that there is not two human individuals identical, even if they have the same genome.
Mm.
Because you can have even right to left kind of polarity of the cerebral cortex in monozygotic twins.
I think we have one question here, one question there. I think Angela, if you want to start, and have we have something online as well? Okay, Angela,
It's
please.
a bit of your main topic, and that was a relief that you only mentioned glia, I think, once,
Glial
Yeah.
cells.
glial cells. You only mentioned it once, and I just wondered what we know about that in size of the brain, in complexity.
Glia.
You know, glial,
Yeah.
glia.
Glia.
Yeah.
Ah, glia. I'm sorry. Yeah, sorry, I did not say it.
Is there anything that you think is important about the glia?
Of course it is. You know, even there is a big contribution of glia in synaptic epigenesis, I did not mention. And it's I'm sorry to say, an additional complexity. But on the other hand, thanks to the adaptability of glial cells, you don't need a lot of genes to have glial cells and to repeat themselves throughout the development. Because in fact, they are not ordering the brain, but the neurons, to my opinion, are ordering.
Well, that's really what I wanted to know, because there is so much interest in the glial cells these days, and there's the synapses they form, and there's a great deal about transmission.
Of course, there is a contribution, and I may say that I should add glia as another connectomic fundamental. I will. Thank you very much for that. But to my view, it's rather simple compared to the development of neurons. That's what I. Now, it does not mean that the role of glia is simple and even simple-minded. In fact, when you have glia at some position and some other one, then the function is different and so on and so forth. So you may have a broad diversity of functions which are crucial and critical, and that should be mentioned. I thank you very much. I will mention it next time.
You didn't say too much about them. The immunologists are very excited about glia.
No, I understand that you might be, and I'm sorry. I feel really sorry for not having mentioned it throughout my talk. This is not, as I said from the beginning, I say I am going to select a few connectomic fundamental among many other ones, and the other ones, the glia is some. But to give back to the merits of glial cell, I will introduce them as a connectomic fundamental. If you,
I'm very happy if you say that it's the neurons that really count and the glia are just being useful.
No, but I think the debate about glial cell and neurons, I also perhaps a reason why I did not talk too much about it, that there are some defender of the glial theory, like Yves Gilles in France, who say the glia organize the brain, almost. Well, he does not say that explicitly, like I say, but it gives to glia an immense role, and he said, well, I am going to write a book which is a glial human,
L'homme glial.
l'homme glial, compared to l'homme neuronal, which actually he did a very nice book on that. And I may say that I fully agree with what you say, but So what he said, nevertheless, my view is that the main functional aspect is the connectivity of the neural network.
Mhm.
The main one is that it is a network. And
Not
it's
the
not a
individual.
not a network of glial cells, it's a network of neurons.
Dennis, please.
Is you referred in your talk to the form of Darwinian selection.
Yes.
And I remember that I was led from your book on neuronal all of those years ago to look at the work of Gerald Edelman, the immunologist who switched to looking at the nervous system and came up with the idea of neural Darwinism. Is there a connection between?
Of course there is. He quotes us. And I may say he reads French very well. And I gave him Neuronal Man when it appeared in French. He read it, and he told me I read it 3 times.
So his book, Neural Edelmanism,
Neural Darwinism.
as Francis Crick called it, Neural Edelmanism.
Well, Francis Crick rubbished it.
Yes.
Yeah, but I don't think he was right.
No, I don't think it was right either. But it, of course, appeared in 1990. And so I think there's a very clear link.
I think in your question, there are 2 issues. One is a scientific one. The other is the relationship between scientists. And the relationship between scientists, in the last book I wrote, I simply forget about it. I don't even mention it. We are interested by ideas and not by this kind of way. Scientists are very ordinary human beings. You know, they are not specially gifted ethical and aesthetical kind of issue. They are just regular. And therefore they behave, they have this kind of ego conflict. Now, the history is that I wrote Epigenesis in 1973, Neuron Man 83 or 85, and of course we had a lot of discussion, Jerry and myself, and he developed this neural Darwinism following the early work we did on epigenesis, and the one now I think he made it larger brain scale and so on, so that he has his contribution as well. You know, each of us has his own contribution. Let's put it this
Thanks
way.
for that clarification. I'm going to have to leave, unfortunately. I've got a meeting in the university.
Thank you very much for coming.
It's
Thank you.
absolutely delight.
Deeply touched.
Yes.
We'll take one more question and then we'll go now and have some coffee as well. There's one online, yes?
Yes, there's a question from Yin. They say, thank you for the fascinating talk. What do you think confers the sense of self and a continuous consciousness?
Of a sense of touch.
Of
The
self. Self.
Yeah, oh, self. I think a lot about self. But I think it's a very interesting issue. And as far as I am concerned, but there are many different approaches. I work with Hans Lu, who is a common friend from Denmark. And he had some views about self-consciousness from an experimental program he developed with yoga and top-down control of emotion from the cerebral cortex. And he came back with some kind of circuit, and I further developed that with him from a pharmacological and biochemical point of view. Unfortunately, our papers are not so much read. They are not quoted. And perhaps the person who asked the question should read these papers. But I am very much interested by this issue. And I can tell you that here, again, we have a lot to discover. for the future. A lot. And the
Exciting
second
time.
question was what?
There was just a continuation of self over time. So how self continues over time and how it may change. I think, yes. Is that the-
Yeah, confers the sense of self and a continuous consciousness. Is how it is.
Continuous consciousness.
Wonder.
Well, it's, when we sleep, I hope you are not sleeping yet. There is, of course, a loss of consciousness which is taking place. Now, what is interesting concerning the dynamic and so on is when you look at ignition, which is taking place for conscious processing, and the response in V1 or V4 for visual objects. By electrophysiology, you find sharp kind of response. Now, with ignition, you showed much longer lasting response. So there is, to my opinion, some big difference between non-conscious and conscious representations. I did not discuss that at all, but I want to just take the opportunity to say that. And if I may say, the electrophysiology, even though there are a lot of electrophysiologists in the world, but they are not focusing on that. I think it's, to me, it's a mistake, because the most interesting issue for me is the electrophysiological difference between a conscious and unconscious representation. We know that conscious representation have some property. And the most interesting property, which goes along with continuity, is that they can make chains. And that when, in the language, when one object linked to another, to a third, at the end, there is an original output which is taking place, which is the meaning of the sentence. So you have to realize that you have some continuity, through the development of a sentence, where during this continuity, the conscious representation are articulating with each other. How does this happen at the electrophysiological? It's a very simple question to ask the electrophysiologist, but it's extremely difficult to find a way to do it. So,
well, we should continue that. More Prof.
I hope that some of the younger generation will be able to approach. And to conclude, I wish to say that I am very pleased to be here, to have this exchange, and also that each lab is trying to enrich the knowledge at the world scale by its own contribution, and that positive interaction between labs is essential. Multidisciplinarity is a key.
Thank you so much.